Thanks so much. We're very excited today to have Marcus Lundberg, MD, PhD, coming to us from Uppsala University Hospital and Uppsala University. And really appreciate you joining us. It's sort of late there. It's later, 6 o'clock-ish. And so appreciate you coming on and giving this talk because it is really exciting, important. It was featured in our Week in Review newsletter and it generated a lot of interest. The title of his talk is Islet Delta Cell Architecture is Remodeled in the Human Pancreas During Type 1 Diabetes. And a little about Dr. Lundberg. He's a physician scientist affiliated with the Department of Immunology, Genetics, and Pathology at Uppsala University and with clinical immunology and transfusion medicine at Uppsala University Hospital. His research is dedicated to uncovering the pathological mechanisms underlying type 1 diabetes with a particular focus on human pancreatic tissue. He's made contributions to the understanding of islet inflammation, vascular changes, neuronal remodeling, and gene expression alterations in the pancreas of individuals with type 1 diabetes. Most recently, his work has advanced knowledge of extra-islet endocrine cells in type 1 diabetes, and although his primary aim is to uncover the pathology of type 1 diabetes, the current work on delta cells is focused on how the distribution of these cells in the pancreas may influence hormonal secretion and especially the impaired glucagon response during hypoglycemia in type 1 diabetes. I'm going to drop the paper in the chat and feel free to add questions in the chat. We'll also take questions at the very end. Thanks so much for joining us, Marcus.
Thank you very, very much, Monica. Thank you for inviting me. So this paper that I will present, the methods and the results, they are quite straightforward. And they are focused mainly on the delta cells location within the pancreas, but also the connection to other endocrine cells. However, the background is a bit more complex regarding delta cells in type 1 diabetes. I have prepared very, very simplified schematic overview. So it lacks a lot of arrows, intracellular signaling, etc., but I think it will convey the most important points for this topic.
Simple but elegant.
Yeah, I hope so. Yeah, so I start by introducing the alpha cell. So during low glucose levels, the alpha cell secretes glucagon, which causes elevated glucose levels. However, in type 1 diabetes, there is an impaired glucagon response to hypoglycemia. So this is a problem for patients with type 1 diabetes. Hill et al. Made a paper last year, or published a paper last year, where they looked at isolated islets from donors with type 1 diabetes, and they found that they do not secrete glucagon in response to low glucose. So I think it was a very nice study. Basically, with low glucose levels, there was basically no glucagon secretion. And that shows in a very nice way that something is dysfunctional in the type 1 diabetes islets. So here enters the delta cell. The delta cell has several functions, but in this simplified model that I'm showing you here, we focus on somatostatin that is secreted by the delta cell and inhibit the alpha cell. So basically, more somatostatin leads to less glucagon. Hill et al. Also showed in type 1 diabetes islets that when they were incubated with somatostatin receptor inhibitors, so you block this path here, then there was an increase in the glucagon secretion. And I think that's very nice because it shows that in the type 1 diabetes islets, the interaction between these cells are of importance in relation to this impaired glucagon response. And they also examined beta cells or islets from animal models. And again, very simplified, they found that there is an interaction between beta cells and delta cell that causes, in this situation with low glucose levels, there is an inhibition of the delta cell, which leads to less somatostatin, and then the alpha cell can do its job that it's supposed to do during low glucose levels. They also postulated that when the beta cells are lost during type 1 diabetes, you get more somatostatin, and that causes the impaired alpha cell function and less glucagon secretion. So when I made this presentation now, or this schematic illustration, I cited this paper very much. And if you want to read additional papers that are also important in the literature, I recommend you to read the original article by us. But yeah, this article is really good, so I recommend you to read it. Histological information of delta cells in type 1 diabetes is very limited. The proportion of delta cells and alpha cells within the islets during type 1 diabetes has been described in several studies previously. But except for that, there's basically no information. So we thought that if the delta cell architecture is altered in diabetes, this could also affect regulation of hormones. So the aim of this study was to examine delta cells within the islets and also outside of the islets. I will get back to these extra islet delta cells later on because I have a nice image that illustrates what this really is in a better way. We especially focused on type 1 diabetes and also included some type 2 diabetes specimens in this study. Delta cells in type 2 diabetes has been more extensively studied previously. So pancreatic sections from 9 type 1 diabetes, 6 type 2 diabetes, and 30 non-diabetic donors were included in the study. We did a very straightforward co-staining for somatostatin, glucagon, and insulin, and also very straightforward manual examination of cell connections and localization of delta cells within and outside of islets. And when I talk about cell connections, in this case, we defined cells as being connected to each other if they were adjacent to each other, as in this illustration here. If there was a gap between the cells, we counted them as not being connected. Here is an overview image of a scanned tissue section. You see the exocrine tissue, but also in green, we see the beta cells. In red, we see the alpha cells, and in white, we have the delta cells. Here is a zoomed in image. We have some representative images from a non-diabetic donor, from a type 1 diabetic donor, and a type 2 diabetes donor. Here is some of the, or perhaps the main finding of this study. We looked at the number of connected alpha cells per delta cell, and for each donor, which is represented by a dot here, we obtained an average number. So for example, this dot here says that perhaps 1.4 alpha cells were connected per delta cell. On the x-axis here, we see the different groups, non-diabetic, type 1 diabetic, and type 2 diabetic group. And the median number of connected alpha cells per delta cell was approximately twice as high in the type 1 diabetes group compared to the non-diabetic group. So there were more alpha cells connected to the delta cells in type 1 diabetes. The number of connected beta cells per delta cell was reduced in type 1 diabetes. And the number of connected delta cell per delta cell was, we couldn't determine any differences here between the different groups. We thought that perhaps in type 1 diabetes, when there is a loss of beta cells, perhaps the delta cells would become gathered in aggregates. But these findings suggest that that is not the case. However, we also made another analysis to look at the connection between delta cells in parallel. So for each islet that we examined, we determined whether the delta cells were mainly single cells or if they were mainly in pairs of 2 cells interconnected to each other, or mainly in 3 or more cells interconnected with each other. And with this semi-quantification, we couldn't find any differences between the groups either. We also made a semi-quantitative analysis of the location of the delta cells within the islets. So we determined whether they were mainly in the core part of the islet, whether they were equally distributed throughout the islet, or if they were mainly in the peripheral part of the islet. And on a group level, there was the type 1 diabetes group was prone to having the delta cells in the peripheral part of the islet, a slight difference. So the last result is regarding the extra islet delta cells. We defined extra islet delta cells as being single cells in the exocrine parenchyma without any connection to other endocrine cells. We looked at this because in a previous study by us from last year, we found that the extra islet alpha cells were 5-fold increased in type 1 diabetes. In the current paper now, when we looked at these cells, they were very rare in general, but they were more than 3-fold higher in the type 1 diabetes group. It's quite difficult to speculate on what this actually means and what the impact of this change is in type 1 diabetes, as there is no literature about these cells from before. At least I haven't found anything about them.
Marcus, did you, or maybe you didn't look yet, but is there any evidence to show that those extra islet delta cells might have connections to macrophages or leukocytes?
We haven't done any such analysis in this study. We haven't stained for leukocytes or macrophages.
Just wonder if they were like some kind of intermediary there. Why are they doing that?
Yeah, so you're starting thinking about what's causing these cells to get there. Yeah, no, we don't have any clear speculation about what's causing this. This is the density of kind of cells. And we know that the volume of the pancreas is reduced in type 1 diabetes. And if this volume reduction is mainly due to a loss of acinar tissue and that these delta cells are not lost at the same time, then we would get a higher density due to that. But we don't think that change, that volume alteration, would explain this quite high fold change that we found here. So yeah, we're very curious about these cells. Before I get to the conclusion, I will just summarize the results. So there was an approximately 2-fold higher number of alpha cells directly adherent to delta cells in subjects with type 1 diabetes compared with non-diabetic subjects. The delta cells were more peripherally located within the islets of donors with type 1 diabetes. The density of extra islet single delta cells in the acinar region was more than 3-fold higher in type 1 diabetes compared with non-diabetic subjects. And last but not least, something which I haven't really mentioned previously in the presentation, is that there was no difference in delta cell architecture that could be determined in type 2 diabetes compared to in non-diabetic subjects. So a general assumption is that when a receiving cell is closer to a secreting cell, the receiving cell will get more exposed to the secreted compound. So in this kind of model that I made here, if the alpha cells are closer to the delta cell, they might be more inhibited and then secrete less glucagon. And alpha cells that are further away might be less inhibited and secrete more glucagon. So based on this, we wrote that a higher number of alpha cells directly adherent to delta cells in type 1 diabetes likely increases the alpha cells exposure to somatostatin. This finding may be a link partly explaining the reduced glucagon response to hypoglycemia in type 1 diabetes. However, I want to stress that we only have descriptive findings in our study. So even if we believe that this may be of importance, we can't say how large an impact this actually has, the distance between delta cells and alpha cells. I want to finish by mentioning some limitations and future perspectives. On the delta cells, there are filopodia, basically protrusions, and they are connected to beta cells and alpha cells. Because we examined only thin sections in this study, we couldn't examine the filopodia. But I think it would be very nice to make a study where the filopodia were also examined, because it's like a whole dimension in the connection between endocrine cells, between delta cells and other cells, that we can't really say anything about in this paper.
Marcus, in live slices, have there been imaging of filopodia and, in movie settings?
In live slices?
Yeah.
Yeah. Have you read that, or
I
is it?
haven't seen, no, I
Yeah.
don't know. And maybe
Uh,
someone in the audience knows. There's a couple of questions holding, but
Yeah, the paper that I think reported this in a very nice way is the one that I'm referring to here. They have examined Philopodia, and I think they used some, I don't remember exactly how, but they had to treat their tissue in a special way to make it possible to look at the 3D structure and really study these Philopodia. I'm not sure if it would be possible to do it in slices. I can't really answer that. But that would be very interesting if it's possible.
yeah, maybe we can get some light sheet going. Who knows? Okay.
Yeah. Image analysis using different softwares, I think, will provide additional important information regarding the delta cell architecture in type 1 diabetes. We did this quite simple analysis, but I think there's many dimensions here that we haven't really been able to report. And again, I want to stress that the paper is based on descriptive findings. So additional functional studies are required to really know how the distance between delta cells and other endocrine cells affects the hormone regulation.
Yeah, well, it's really, really a great paper. We have a bunch of questions in the chat. Alexandra Ripper, do you want to unmute yourself and ask your question?
Hello, everyone. Yes, my question was about denominator. When you count interactions in between alpha and delta cells in islets in type 1D versus controls, your denominator will be islet area because there is a variance in islet sizes. I assume in larger islets, you will count like more connections. My questions specifically, what was the denominator, and could you like comment on islet size?
Yeah, so we wrote that also in the paper, but I didn't mention it here. But the average islet size was basically the same, or there was no significant differences, at least, between the different groups. But we haven't been able to really look at that dimension either, if it's different in small islets or differences in large islets. But at least we don't have any like that differences between the groups.
Yeah, it'd be interesting to see that. We talked a little offline about the fact that Sarah Richardson's group recently uncovered the existence of these endocrine objects. And it's just a really fascinating new advent into the study of type 1 diabetes, these smaller, almost more fragile looking endocrine objects. And she's brought that to bear. So her group's looking closely at that. Might be interesting to look at what the role of delta cells is in the smaller sized precursor setting as well. Okay, Cassian, INI in diabetic islets, do delta endothelial cell contacts degrade, persist, or adapt in ways that correlate with disease progression or hormone dysregulation? Also, do delta cells influence endothelial cell function? And Cassian, if you want to unmute yourself, or you can answer it.
Yeah, I don't know anything about delta endothelial contacts. So we haven't looked at it. We haven't stained for endothelial cells. So because of that, I can't answer that question. But, and I can say that I haven't even thought about that, but it's a very interesting question. So yeah, do delta cells influence endothelial cell function? I'm not sure about that, actually. I haven't focused on that when I've gone through the literature. So I don't know. But delta cells have many functions, so it wouldn't surprise me.
Okay, Kathy Gerder from Medical College of Wisconsin, have you considered differentiation or transdifferentiation during stress?
Whether the delta cells, I'm not sure if it's a question if we are considering to like do this kind of experiment where we expose delta cells and see what happens, or if it's more question about maybe whether the stress in type 1 diabetes could explain these extra islet delta cells. And to answer the first question now, we haven't made any such attempts to look at that. But we have thought about it like in these extra islet cells, whether the stress could somehow cause transdifferentiation, because we have found previously we made 2 papers last year about extra islet cells where we looked at insulin positive cells and glucagon positive cells. And when we looked at glucagon positive cells in type 1 diabetes, there was a 5-fold increase. So that makes us wonder, like, could there be a transdifferentiation that the beta cells become alpha cells instead? But yeah, it's very speculative, and you could speculate in the same way here as well, that we get this loss of beta cells, and on the other hand, we get more delta cells.
Okay, great. We have a bunch of questions here. David Hill, thanks, Marcus. Is there any data to suggest the increased Asner delta cell number in T1D is due to proliferation?
No, so I haven't found anything about these cells previously in the literature. We haven't performed any staining for like Ki67, for example. So we haven't looked at this. I think it would also it would be challenging to look at because they are very few. So you would need a lot of sections to really find a sufficient amount of cells to get a good percentage of how many that are Ki67 positive.
Maybe pink base with an intersection with AI might be helpful.
Yeah, absolutely.
Okay, so let's see. We've got Ahmed Brida. Is the increase in alpha cell connection due to transdifferentiation of beta to alpha?
Yeah, that I cannot answer. I think it's an interesting question, and many people are discussing like transdifferentiation, whether it's occurring or not. I can mention in that context that when we looked at extra islet beta and alpha cells in our previous papers, we also examined ARCs and PDX1, and we found that many of these cells were lacking these transcription factors, which could indicate, we can't prove it, but it could indicate that they are more immature or that they are in some kind of situation of transdifferentiation. But yeah, that's very speculative, but yeah, that's what we found.
Okay, and Quan Zhang as asking, did you see the difference in different T1D progression and stages, like for instance, long standing or recently onset T1D?
In this paper, we only included long standing cases. So we can't answer that. We can't say whether there's any differences between these stages.
Okay, and then another question from Kiati Girder. Sample size was limited. Did you observe any difference with HLA or age?
With HLA, we didn't look at HLA. That was not available. And with age, no, we looked at the age in the non-diabetic cases, and we couldn't see any like correlation in how, at least not in how the density of delta cells within islets look like.
Well, I'll have to say, you've opened the door here to a lot of really interesting questions and certainly to more inquiry. I mean, people are getting very excited around this and asking, peppering you with questions, but it just sort of shows that this is the first step and that there's a lot more work to be done. Okay, here's a couple more here. Tom Hill, thank you so much for this excellent presentation. Congratulations to you and your team on this work. Are these extra islet delta cells found in closer proximity to the pancreatic ducts?
I think that's an excellent question. In this first analysis, we just looked at the density. So I guess the next step is to start looking at these other factors, where they located to close to ducts or not. I can only say from my what I have in my memory that they were at least quite far away from islets. So I don't believe they were like part of a scattered islet or any such thing. They were found somewhere else in the exocrine parenchyma.
Big question. Where are they going? Okay, and Benoit Hastoy, thanks for this talk. Did you see any correlation with macrophage infiltration?
Yeah, so that's also what asked previously. And we haven't made any stainings for leukocytes or macrophages or any such. So I don't know. I can't answer that.
Okay, well, we're at our 30-minute mark. Marcus, this is really exciting work, and it just shows with all the questions that, as I said, you've opened the door to a whole nother level of inquiry here. So it'll be exciting to see where your group brings it, and we'll be looking for your next paper, and congratulations on this really interesting study.
Thank you very much, and thank you for inviting me.
Yeah, thanks for
And
joining
thank-
us.
Yeah,
Go relax and enjoy your evening.
Yeah, and thank you everyone for great questions.
Thank you. Have a great rest of your evening. Bye-bye.
Bye-bye.